All right, so good morning, everybody. Final day. The topic of today would be tissue engineering and what we have established so far in the lab to provide silk as a scaffold for any type of tissue engineering. So what I will talk about is a little bit of, giving you some background about wound recovery. So how do wounds heal? Then I will show you some properties of tissues and also properties of scaffolds. Give you insights into cells on surfaces. So today it will be also a little bit more biologic than in the last few days. And then I will show you some quantitative applications. So what happens when you damage any kind of tissue? I mean, in this case, it's shown for skin wounds, but it's also true for any kind of other wounds. Typically, wound recovery is a process that has three different phases. The first phase we call the exudative phase. Exudative phase takes place between day 1 and day 4. And in this exudative phase, actually all substrates, all dead cells, everything that doesn't belong there in a wound is removed. So therefore, we also call it the catabolic phase. So here's the wound, and you see there are some dead cells in here, and everything has to be thrown out and removed. And because, of course, we do have also damages to blood vessels, there would be bleeding. And in order to avoid that we bleed to death and lose all the blood, of course, we have to clog somehow here the open side of the wound. And this is actually done by proteins, mainly by proteins. The most important one is fibrin. Fibrin will be cross-linked, and this cross-linked fibrin together with some special cells, the thrombocytes, will actually make the clog and close the wound. Of course, there will be an inflammatory response. Inflammatory response actually means that phagocytes will ingrow, and these phagocytes, they're special cells that are necessary to remove leftovers and also to remove bacteria. So the phagocytes then will phagocytose microorganisms, mean they just, it's like they eat the bacteria that are around there, but also other particles. And so this is one of the mechanisms to remove stuff by phagocytosis. Then finally, the capillary network will expand to improve the blood circulation surrounding the wound, but this only starts to work once the cap is on, because if not, I mean, of course, it would be counterproductive if you bring more blood to a wound and it's still open. And then finally, when the big stuff is removed by the phagocytes, some proteases and lytic enzymes will be released, and they actually now take care of other proteins, of DNA, whatever is else found in the wound to remove that. And this, of course, is a very critical step if you talk now about using any kind of scaffolds or whatsoever. If we talk about silk scaffolds, right? They're made of protein. There's two ways. I mean, if we have like particles, they can be uptaken by phagocytosis and removed. So that could be an issue. And the other issue could be that the release of the proteases in the wound could just degrade the scaffolds. And then, of course, the scaffold is gone. So this is something you have to take care of. And what we did, we developed an in vitro model in the lab where we actually can mimic all the proteases, the whole mix of proteases. So there is a collagenase, and there are different metalloproteases that are secreted here. And we have that in the lab, so we can mimic this wound solution and can test then scaffolds inside that wound, if they're stable or not, for instance. So this goes on for the first 4 days, and actually there's nothing to accelerate that. So even if you take drugs or do any kind of treatment, you cannot accelerate that phase. No way, whatever you do. That's very important to know. Then the second phase is the so-called proliferate phase, and then it starts on day 5 and it goes on until day 14. So this is a real anabolic phase. Now, some stuff will be rebuilt. So after removal of the stuff for the first 4 days, after 5 days, the tissue starts to regenerate by the proliferation of capillaries, the formation of vascularized tissue. New cells grow in, and these are especially so-called myofibroblasts and fibroblasts. Fibroblasts and myofibroblasts are specialized on synthesizing collagen. And collagen, of course, will be needed to make a new scaffold here, a new tissue, because collagen is the major component of the extracellular matrix. So the stuff that surrounds cells, that surrounds also capillaries. And actually, therefore, we need these cells, and they're directed here. You see here some myofibroblasts in there. They're directed to the wound in order to do the synthesis right at the hot spot, so to say. And again, this is a phase, the proliferous phase, you cannot speed up. So typically, if you have injured your skin, have a cut, it always takes like 2 weeks. You cannot speed it up, whatever you do. It takes 2 weeks, until these phases are over. The only phase that can be accelerated then by using creams and drugs and so on is the third phase, the so-called repair phase. It starts after day 15. It can go on for months, even for years. Sometimes even after years, the repair phase has not been done. It's a matter of fact that still differentiation goes on, maturation, so there's a remodeling going on in the tissue all over time. And of course, you see then scars here, some defects on the outer layers, but the same is true inside, and there's instantaneously remodeling going on. So all the wound defects, like the fibrin clogs and so on, they will be replaced by collagen bundles, and then a new tissue will be formed, which actually has not this high density of capillaries and cells, so the cells have to be removed again. The capillaries that have been ingrown in the first place have to be removed again. And in order to remove these cells, they're again taken up by phagocytosis, and now these phagocytosis is done by a different cell type. It's called the histiocytes. So they're specialized to remove fibroblasts and so on. So as you can see, a very complex phenomenon here. You see these histiocytes, they're actually uptaken here, fibroblasts. So quite a complex process, and as I said, this takes quite some while, can take up to several years. And this is something you can support by additives, by having scaffolds around, having drugs around. And this is actually the phase that we're looking for. So of course, there is some acute wound recovery, meaning that, of course, we can help wounds here to have a better shielding, that no bacteria grow in, and that actually this closing of the wound works better. This is something we can do, but this has nothing to do with kinetics. This is more something like about having better equilibria. But then the other topic of tissue engineering is then to actually improve the repair phase and make the kinetics of the repair phase faster. So far for the background. Now, something that is, of course, of high importance if we talk about tissue engineering, it's about mechanics. And this is, again, something you're very familiar with. And here's some mechanical data of typical tissues. Cartilage, liver, and then 2 layers of skin, the upper layer and the lower layer. And as you can see, we have very low E-moduli. We're here in the kilopascal regime. So cartilage is sort of like the stiffest with between 100 and 200 kilopascal. But liver, for instance, has a very low E-modulus, 1.5 kilopascal. And skin, even the upper layers, they have even a lower E-modulus. This is very important now to know, because if we're talking about tissue engineering, we also have to consider that if I use a scaffold, we have to be somewhere in the range of these E moduli. So if we talk about using spider silk fibers, let's say, for skin recovery, you see that we are here off by orders of magnitude. So whenever I read the papers, coming from some medical groups, that they do skin recovery with silk fibers harvested from the spiders, mechanically this doesn't make any sense, because how should such a fiber support skin growth? I mean, it's much too strong. There will be no possibility for the surrounding skin to deal with that tissue, because there is a lot of repair going on, but how can you deal with something that is 100,000 times stronger than you? You won't touch it. So there will always be a break between the surrounding tissue and the ones where you place the fiber. It's something different if you want to have guidance for cells, nerve cells, because they just really need the fibers for guidance. But if you talk about tissue engineering, skin engineering, silk fibers, as harvested from spiders, they're way too strong. Doesn't work. So therefore, you have to think about probably other types of processing, and I showed you several processing types that you can do. So you have, of course, to regenerate either the silk fibers or take biotechnologically produced silk proteins, and then you can probably make hydrogels, and you see at least we are here in a better ballpark, or you just freeze-dries the silk, and then we're at least here in the lower megapascal or even in the kilopascal regime, but still the mechanical properties here are much too high. So if you think about making liver tissue, even these hydrogels and even these scaffolds are much too strong. So that won't work. It could work here for skin recovery, but also just for the lower layers, not for the upper layers. This is something you have to keep on mind. Then, of course, as I told you, it's very important to know how stable a scaffold is in such environment. And I told you, I haven't have the data here, that we already developed now an in vitro model where we could really mimic an entire wound. But normally, you just start with simple model systems, and this is just a set of enzymes that have been tested. Trypsin, alpha-chymotrypsin, collagenase, protease 14, proteinase K. And what you can see here is that at very low enzyme concentrations, you do get, well, with some proteases, some sufficient degradation, even within a few hours. Of course, proteinase K is a very unspecific protease made by some bacteria. This is typically nothing that you have to deal with if you work with wounds. Enzymes in wounds are very specific, and they degrade probably the scaffolds much lower. But proteinase K, for instance, was the one we used to dissolve the capsules to see if, in principle, we can digest the proteins. Typically, you have something like collagenase 1A or protease 14, and as you can see here, the degradation takes several days, and in the case of collagenase 1, it's not even complete. Like after 15 days, we still have 50% of the silk molecules intact. And there's even other enzymes like trypsin. After 22 days, we only have 5% of degradation. This indicates that silk scaffolds are quite stable against some enzymes, while they're not against others, but, well, the ones that probably very well digest the silk molecules are highly unspecific and typically not found in the human body. Of course, if you take soluble protein, all of these enzymes will digest the protein within 2 hours. So this is really a scaffold. But it doesn't matter if it's a fiber or a particle or whatsoever, because the structure is literally the same, right? I mean, that's what I've shown, that the secondary structure in a particle is identical to the secondary structure in a film, is identical to the secondary structure in a fiber. Just the arrangement of the structure is different, but for the enzymes, that doesn't make any difference. Okay. So this indicates that silk proteins are suitable. So we have quite often intermediate or slow biodegradation, at least in some cases. I've shown you already that we can make drug delivery devices, so we can load and release drugs with high efficiency. We do have good mechanics. For most cases, the mechanics are too good, so we have to actually lower the kinetics for some of these tissue engineering activities. And then, of course, there are 3 more questions. How about toxicity? How about immunoreactivity? And how about cell interaction? Toxicity, well, this has been tested also in animals quite often now. It has not been seen any kind of cytotoxicity, meaning no toxicity on cells, and also no acute toxicity, which is the toxicity related to a whole animal. So silk proteins in the soluble form as a scaffold or even the natural silk does not induce toxicity. It's a well-tolerated protein, so that's good to know. I already mentioned, I think yesterday, a little bit about immunoreactivity. Well, if you have natural silk proteins, it's very hard to get antibodies against it, and to my knowledge, there has only been one group, Axel Sponner, who has been able to raise antibodies against some terminal domains of silk proteins. We haven't been able to make any antibodies against the silk sequences, so the immunoreactivity seems to be low. You have to be careful if you work with biotechnologically produced proteins. As I said yesterday, typically you have tags on it for purification or for whatever reasons, and these tags can cause immunoreactivity. So before you bring a silk scaffold made of biotechnologically produced silk into a human body or into an animal body, you should be very carefully removing the tags so that you do not get immunoreactivity. But I'll show you later some examples where we've tested in rabbits and especially in rats that there's no immune reaction even after a year. Now the final question is cell interaction, and this is a very important one because the big question, of course, is how do cells respond to the presence of silk, the proteins, the surfaces, the topics, and this is what I would like to highlight now in the next few minutes to show you, to give you some insight into how cells interact with surfaces, what is important, how do silk surfaces interact with cells, and then how can we actually play around and improve or deprove actually the interaction between cells and the scaffold, and what that could mean for possible applications. So what determines the adhesion of a cell to a surface? I mean, there's literally 4 major things you have to be aware of. It's the topography of the surface. It's the mechanics, especially the elasticity of a surface. It's the wettability, so the hydrophobicity. Of course, if it's very hydrophobic and water can't spread very much on a surface, then also cell interaction is low. With elasticity, it's the same. So cells like stiffer materials more than elastic ones. So if you have a ceramics and a soft polymer, they rather grow on the ceramics than on the soft polymer for whatever reason. And then topography. Of course, if you have a very flat surface, it's very hard for them to grab onto. So you need some, microscale features that they can anchor to so that they can hold on the surface. So these 3, of course, are important, but I would like to focus now on chemical signals like ligands, because these chemical signals we can nicely control. I mean, we also can control the other features, but chemical signals, they can be very specific for the type of tissue. The first 3 points, they are valid for all kinds of cells. If we talk about skin cells or bone cells or muscle cells, it's all the same. I mean, here with these 3 properties, you can actually manipulate all types of cells to surfaces. Chemical signals, they are specific for a certain tissue. So skin cells, keratinocytes, and fibroblasts, they actually like different types of chemical signals than bone cells, than muscle cells, and so on. So with this trick, actually, you can manipulate and you can actually attract a specific cell type, and you can differentiate between different cells. So and therefore, I would like to focus on that a little bit more. Chemical signals for cell adhesion. What could be chemical signals from artificial surfaces? And of course, if you take a silk fiber, put it to the body, it's an artificial surface. Even if you make anything else, I mean, it's an artificial surface. And you can now passively or actively put chemical signals on top. Passive chemical signals would be any type of protein that adsorbs to a surface, because protein adsorption actually conveys cell adhesion. This means a cell can never attach to a surface directly. First, there has to be a layer of proteins, and cells always attach to proteins. So if you have a surface that shows a very good unspecific protein adsorption, you also have a good cell adhesion. And this is what we call passive, because it doesn't matter what type of protein you take. And therefore, again, this is true for many types of cells. There's no specificity. Therefore, it's a passive chemical signal. And then, of course, there are active chemical signals, the ones that I've already mentioned. Bioactive ligands. This can be proteins, that can be peptides, it can be whatever, that are very specific for individual cell types. And these bioactive ligands, of course, they differ from tissue to tissue. And if you bring these ligands to an artificial surface, you have some active response because you get specific cells down to the surface. Let's start with the passive induction of chemical signals. Well, cells specifically recognize interfaces, meaning a surface. And if the surface is attractive, a cell absorbs. But it's never, as I said, directly adsorption to the surface. Cells typically adsorb to proteins. So therefore, actually, it's a matter of fact if a surface or an interface is attractive for proteins. If it's attractive for proteins, it will be attractive for cells. So proteins that interact with the surface, of course, make it then attractive, and these proteins can even be produced by the cells themselves. So it could be collagen, for instance. So a fibroblast swimming along a surface is producing instantaneously collagens. If a surface is then attractive for the collagens, the fibroblasts will have some grip, and they can hold on the collagen, and then they produce more collagen, and this is how everything starts. So the most important question here is now, is a surface attractive for proteins? So this is a very simple test. I mean, you take something that you would like to use as a cell culture plate, any kind of material. Typically, this is plastic, but it can also be metals or whatsoever. And you just put your culture media on top. And of course, in a culture media, you do have quite a lot of proteins. And if you have a good cell culture plate, actually you have a good protein adhesion. This is very important in order to grow cells. So there are also sort of negative controls for cell culture, so plates that do not show good protein adhesion, and you always need that to show that you really have a specific effect that cells grow on your surface. And typically such a very low-performing surface would be polystyrene. Polystyrene surfaces, they do not absorb a lot of protein, and therefore cells do not grow on polystyrene. So as soon as the proteins are down to the surface, then of course the cells are able to produce, to attach to it first weakly, then they produce collagens, and then they of course adhere much faster. And the reason why they adhere faster is because they have very specific receptors. These are transmembrane proteins. They're called integrins, and they can actually now specifically bind to certain sequences in collagen or in other extracellular matrix proteins. So this would be the cytoplasm, so the inside of the cell, and here's the outside. And as you can see, you have here transmembrane proteins, these integrins, and they actually detect and attach to specific extracellular proteins. And importantly, the integrins span the plasma membrane. And this is therefore important because inside, the integrins interact with the cytoskeleton. So with actin fibers, with tubulin, and so on. So the cell can actively then respond by movements to such surfaces once an integrin finds a binding partner, finds a ligand, because then the integrins inside the cell will also coordinate the cytoskeleton and the orientation and the shape of the cells. And we will see pictures on that. Now, just to get some idea about adhesion, I will show you some polymers and their interaction with cells. So this is a very common polymer used for cell culturing. It's polyethyl oxazolene. So you see here, that's the monomer, and this is just polymerized n times. And this is just a picture. It's a 10 times magnification of the cell culture dish. And what you can see here, literally, I mean, this is a confluent cell culture, meaning you have millions of vital cells, and they just cover the surface as good as they can, and they make a monolayer on top of this plate, and you even can't see the plastic anymore. I mean, all you see here is just cells at very high density. So this is a surface that is well suited for protein absorption and for cell adhesion. And now that's the opposite, and you know that polymer, polyacrylamide. Here's the monomer, n times polymerized. What you see, this is even a 20 times magnification. There's not that many cells. I mean, this is the polyacrylamide. You clearly see it, and this is one of the cells. So there's just single cells, very rarely distributed and randomly distributed on the surface. And as you can see, they have very abnormal morphologies. They don't feel wealthy. They don't like the surface very much. So there's only a few of them, and the few that are there, they don't like it. So this is a clear example of a bad surface. So the question is, well, how can we now play with it a bit? And of course, the better would be to have not synthetic polymers, but natural polymers. And the natural polymer that is used often is chitosan. So this is again the monomer, and it's polymerized. So it's a sugar that's a derivative of chitin, and chitin is the sugar in the shell of like prawns and crabs and so on. So you can actually remove chitin from prawn shells and then just chemically modify it. And they show a very nice behavior because cells like the surface quite good. Well, we have some low cell adhesion, but they still, if they're binding, they grow very well. But the problem with chitosan is it's not very stable, so we often have delamination. So you just, layers of chitosan just lift off. And you see an area here where just the chitosan have lifted off, and of course, then if the chitosan lifts off, it takes the cells with it, right? So although chitosan as a natural material has some good properties, it's not perfect because of the delamination. So, so far about the materials. So as you can see, we have polymers in all ranges, good binding, intermediate binding, bad binding. So we can play around with the binding adhesion. Then, of course, we can play a little bit with hydrophobicity. And this is just a very simple setup to show you what cells like and what they do not like. So this is just our substrate. And here with lithography, what we made, put on hydrophobic blocks, and then in between we just have a hydrophilic surface and a hydrophobic block again and a hydrophilic surface. And again, the importance is areas that are attractive for proteins are attractive for cells. So the point here is that hydrophobic surfaces are not very attractive for proteins. So therefore, polystyrene is a perfectly non-cell and non-protein binding surface because it's very hydrophobic. The other extreme, very hydrophilic surfaces are also not very protein attractive. Why? If a surface or a polymer is very hydrophilic, it will uptake a lot of water. It will be swollen. And if it's swollen, there's a lot of water, and the water then, of course, competes with the polymer or with the protein to bind to the surface, and therefore the binding on very hydrophilic surfaces of proteins is also very weak. So this means hydrophobic is no good, very hydrophilic is also no good. So you have just a very narrow window, so medium hydrophilic or medium hydrophobic, that is attractive for proteins and therefore is attractive for cells. And then, of course, it's important if you have charges on the surface, because charges on surfaces will have an influence on protein binding and therefore on cell binding. And this, again, is very important if you think about using silk as a scaffold material. As I told you, silks are amphiphilic. That sounds good. They're not extremely hydrophobic. They're not extremely hydrophilic. They're a little bit of everything. That's good. Some of the silks do not have charges, or not many charges. It's also good because charges will influence protein and cell binding. And this is just an experiment to show you that you really can play around with these properties quite well, and you can actually make structured surfaces. As I told you, if you have a surface that's hydrophilic and you bring on something hydrophobic, like here, you could use, for instance, polystyrene, and a monolayer of polystyrene will be sufficient. Just need like 2-3 nanometers of polystyrene on top. This will prevent the binding of proteins and also prevent the binding of cells, as you can see here, or in the larger scale. So these squares, they're actually patterned with polystyrene, and polystyrene is so hydrophobic that the proteins don't go down and also the cells won't go down. And everything in between, where there is no polystyrene, which are here the yellow bars, there you do see cell binding because there the proteins have bound beforehand. If you have the blank surface, which is hydrophilic in this case. So what we could do, of course, now, we can directly pattern such a surface with proteins. So we can specifically deposit proteins using, for instance, soft lithography. This is a side view, and this is a top view, and we can actually make lines of proteins. So the idea, for instance, is, and this is what we do in the lab, if we just, make a pre-structured line just of silk proteins, for instance, can we grow cells specifically on these protein lines? And here are some images showing that the cells, in this case, these are fibroblasts, that they orient along the chemical signals. In this case, these are just passive signals. These are just proteins, just silk proteins. So they can actually have now a nice orientation only where the protein has been deposited, but not in between. In this case, of course, the substrate is hydrophobic to avoid then any unspecific binding. But this can be done, and we can make nice patterns out of that. So passive induction of chemicals, of chemical signals has some advantages. It's often not necessary to functionalize the surface, right? The experimental setup is simple. And there is microstructuring possible, so we can actually direct the cells to the surfaces and give them orientation. The disadvantages are that, while we have no control on the protein structure that binds onto such a surface, but the cellular response depends on the protein structure, as I've shown you with the integrants. So if we put just collagen in any kind of orientation on a surface, a cell can attach to the surface, but if it cannot find the specific sequences and ligands, the cellular response, the metabolism of a cell, will be different. So this is a disadvantage. If we cannot control the orientation of specific side chains in these proteins that are bound to a surface, we will have no control on the cellular response. And of course, proteins can denature on a surface, so this can have some negative results concerning the cell response. And of course, if we have non-covalently linked proteins, or like this chitosan, they can desorb, and the cells with them. So we have these delamination that I've shown you with chitosan. But the same thing can happen with proteins. So this is sort of like the very simplistic view, and many people do that because it's easy to do. But I think the disadvantages are so overwhelming that this is not a route that we should go for if we want to do tissue engineering. We want to be specific. We want to have control. It's the same as with processing. We want to have control over the cellular response, at least in some dimensions. Of course, you know, controlling cells is difficult. It's a biological system, highly dynamic. And we want to have a control, of course, on the surface properties. So in order to do that, what we will have to do is get some active induction of chemical signals. And therefore, we need now active ligands that, for instance, can be seen and bound by these integrins. So we would like to attract individual cells to a surface. So what we have to do, we have to know the sequence, the peptide, for instance, such an integrin is binding to, and we can mix it with a polymer. So we can either covalently link it and then make a coating on such a surface, or we just make a surface and then later on attach the peptide to it. There are advantages and disadvantages of either route, but the outcome of both will be a surface that has a peptide presented to the environment, and cells can now actually detect these peptides. And if they're specific for one cell type, actually you can even differentiate binding of fibroblasts to keratinocytes and so on, because all the cells have slightly different integrins with different specificities. So by changing the sequence of the peptides bound, we can change the cells that adhere more likely to a surface. So one of the most common sequences is the sequence RGD, arginine glycine aspartate. So it's a natural occurring sequence in several extracellular matrix proteins like fibronectin. And you can use this RGD peptide, for instance, to make now this polyacrylamide, and you've seen it's a very lousy surface for cell adhesion. You can attach now this peptide to polyacrylamide to make polyacrylamide more attractive to cells. And researchers here did a very specific trick, because in nature, this RGD peptide has a very specific structure. And I mean, you're now almost experts in protein structure, so you know protein structure is important. So in fibronectin, the RGD is on something that we call a beta turn or a loop. So it looks pretty much like that. So it's not a linear structure, but it's a structure in a turn. And this turn is necessary because the integrin has a binding pocket that directly fits to that loop. Meaning if you have the whole thing linear, the adhesion would be much weaker. You can imagine if you have the RGD now linear and you have a binding pocket like this, there you have less adhesion than in this case. And for that reason, researchers actually developed something like a cyclic peptide. So here you see the R, the G, and the D, and they're actually circularized, and you can do that by chemical synthesis. That works quite nicely. And here you're now somewhat mimicking this turn-like structure. So therefore, people typically use cyclic RGD. And cyclic RGD peptides work much better in combination with the polymers. So let's see. Yeah? Yeah, so you just need these 3 amino acids. So they're the one responsible. And you see here, you have, because of this circular orientation, you have this kind of, non-linearity. Well, pretty much. It's mimicking it as best as it can. I don't know about the structure in fibronectin. Well, what the people just did is, they just compared the ring structure to the linear structure, and there you see the clear difference that this has a much lower adhesion to integrin than this one has. They did this by surface plasmon resonance measurements and so on. But they did not play, I think they tested different types of rings. Of course, you can make the ring bigger or smaller. And so this is supposed to be the best performing cyclic peptide. But there have been hundreds of studies on how to make that. But I'm not an expert in that. And here you see now the influence of the RGD peptide on cell growth. So that's the polyacrylamide surface again. And you see there's just a little bit of cells. And you see also the cells come in aggregates. So these are not single cells. That's one indication that they don't feel well. And also they're still spherical. And spherical mean also they don't feel well. Because if cells do like a surface, they start to spread. So they try to get as much contact to the surface as they can. And here you see now the difference with the RGD peptide, a much higher density of cells, and some of them already start to spread, and we do not see these large aggregates. Of course, now the cell binding is directly depending on the concentration of the peptide, and here you just see the dependency of a concentration of peptide to the cells we find per area, so cells per square centimeter, and you see there is sort of, at least in a certain regime, a linear dependency of RGD peptide versus cells. The more RGD you have, the more cells you have. But this will not go on forever. If the density of RGD is too high, then you get again a drop. So if you have too many RGD molecules, it's counterproductive. So you need to find like a very specific RGD concentration in order to make it work. So if we talk about peptide-functionalized surfaces, again, advantages and disadvantages. Advantages, simple synthesis. We, of course, can use now a large variety of polymers, because we even can use polyacrylamide that's shown to be very improper for cell adhesion. Now we can functionalize it specifically, and we even can use polyacrylamide. Of course, again, microstructuring is possible, and we can direct the cells to the surface. The disadvantages are, again, that it's very hard to control the peptide concentration on the surface, and also the orientation, and this is a little bit according to your question. I mean, even if you have now these cyclic peptides, you have no idea if they're sticking out straight or if they just bend over, and of course a bent over structure can also not be attracted and bound by the integrant. So this is very, very hard to control. So maybe one more slide and then we make a break. So the point is, and I've shown this already before, the point is now we want to use silk as a scaffold for cells. And well, we can use silk as it is, and I will show you some features for that. But to take some results in advance, silk is a very lousy surface for cells to attach to. And the reason for that is that if you, for instance, make a film, the surface is so flat, and I showed you surfaces of films with roughnesses in the regime of 1.5 nanometers. It's too flat for the cells to grab onto. And also, a silk surface is not very attractive for other proteins. We call that also anti-fouling surfaces. So no other proteins will stick to the silk. And the cells do not stick to the silk proteins. So therefore, if you want to have cells attached to silk, you have to modify either the topography, but then this is again an unspecific cell attraction, or specifically by functionalizing it. And I showed you how we've done that with incorporation of cysteines. So we can incorporate a cysteine, and then we can actually label the cysteines. For instance, with the RGD peptides, we can do that in solution, and then make films, for instance. And then, of course, we have no control over the orientation, but you know, this is doable. But we also can first make a film, for instance, and then do the labeling. And then we even can get like double labeling. We have an anisotropy, so we can actually label the different sides differently. And this allows us some control over the system. So this is one possibility, chemical cross-linking. If we talk about peptide sequences, there is, of course, another possibility. We just can directly incorporate that into the silk proteins. And this is shown here. So these are the two possibilities that we do have. Again, we have our engineered silk protein, 16 times the C motif. And as I've shown you, we can add a tag, for instance, containing a cysteine, and the thiol group can be used now to actually couple such a cyclic RGD peptide. The advantage here is now that we have a very precise control over the amount of cyclic RGD that we put into the system. Because we have a single thiol group per monomer, we can actually quantitatively label monomers, and now we can mix them with non-labeled, with non-functionalized proteins. And we can make now ratios and see how much RGD do we need, 10%, 20%, and so on, in order to achieve the best functionalization. That's one route. The other route, because, well, we have a protein system, we can also incorporate the sequences directly. So we can make a linker that does not contain cysteine, but it directly contains the RGD. And there, we actually speculate, if we, well, do that properly, that because it's a protein, that it makes a better turn structure like we find in the natural protein. And this might then work even much better than this artificial system here with a chemical cross-linker, where you have to be very careful, you know, having the right cycle, having the right linker, so the length of the linker is important. As I told you, hundreds of studies have been done on that, you know, and well, if you test the systems, they're all, you know, more or less fail or don't work that much. So we wanted to see the differences here. And with this, I probably make the break, and I tell you that afterwards. Okay. Are there questions? What we've heard? No. Well, so far we haven't used growth factors because this is not just very simplistically, you know, to adjust the adhesion. As soon as we go to tissue engineering, then we will, of course, I mean, we're working, for instance, on skin remodeling. Then, of course, you will need growth factors to attract the individual cells to the individual layers in a three-dimensional system. I mean, so far, I've just talked about two-dimensional systems, and that's different. As soon as you go to a three-dimensional system, you need growth factors. Okay, let's have a break. Okay, so welcome back. Final lecture. I would like to show you now in the final steps what we do with our biotechnological silk. And here you see already the first examples. What we try to do, we try to functionalize silk molecules, getting them additional features, either by chemical linkage or by genetic linkage, in order to specify attachment to silk surfaces. And here is just an overview of what we did. And this hopefully will be published soon. These are RGD-modified silk surfaces. Why is it necessary to modify a silk surface? Here you see a surface, a film just made of this E80F4, C16. And you see pretty much similar things as we've seen before with polyacrylamide. The cells, well, don't like the surface very much. They do form aggregates. And so this is after day 1, after day 3, and after day 7, and they even detach. So they go to the surface, they don't like it very much, and then they just get released. So if we want to make tissue engineering, we would like to get the cells there. So this surface is not very good because it's not attractive for cells. If we now modify the surface chemically, with this cyclic RGD by using the cysteine mutant, we see a much better adhesion, and we see no aggregation. And you already can see the cells start to spread, and after like 7 days, we have a confluent cell culture here. Confluent means it's covering almost everything. This is the chemically labeled protein, and here's the genetically labeled protein. You see not much of a difference at day 1, but at day 7, it even has a higher density with the genetically added RGD sequence, meaning that we did a good job here incorporating the sequence probably in a manner that allows a native-like structure, and therefore the cells like this surface very much. At the same position where it's here, right up front. Of course, it's very important if you do biological experiments to have proper controls. You can get, you can fail quite easily. So therefore, we did very proper controls. And the proper control is to exchange aspartate with glutamate. It's the same charge, but the side chain of glutamate has one CH2 more than aspartate. And this means if this is just one CH2 longer, it won't fit in the binding pocket. So it's a very specific control. So RGE sequences don't fit into the integrin. Same charge, so you don't play around with anything else, just with the size, and then it won't fit in. Of course. Yeah. And here you see the experiments with RGE sequences, either chemically coupled or genetically incorporated in the sequence, and both show the same results as with in the absence of this sequence. Spherical cells, they make these aggregates, they lift off again. So this makes us very confident that it's highly specific to the sequence, RGD, what we see here, the cell binding, and it's highly specific to the structure. So this, I think, is a very nice experiment showing you that we can specifically functionalize silk surfaces. And I have to say, this here has been done with fibroblasts. So fibroblasts have been the cells we cultured thereon. Now, of course, you can not only do that with films. And as I told you, films have the disadvantage that they are flat. No roughness, therefore the cells cannot bind very well. So we can play, of course, around with the topography. And the easiest thing is just not to use films, but to use a topography that is structured on its own. And the best one here are the nonwovens. I've shown you the applications of the nonwovens for the filtering systems, and we can use the same nonwovens here for wound coverage devices. And this is the experiment. You've seen that picture before. We can control the diameter of the fibers, and by this we can, of course, control the topography. And the number down here just gives you the diameter of the fiber. 150 means the individual fibers have 150 nanometers in diameter. 250, 480, 680 respectively give you the diameter of the fibers, and you can see here that there is a strict dependency on the fiber diameter. Fibers with roughly 700 nanometers in diameter show a very good cell adhesion, and it's as good as here the control, and the control is a tissue that is optimized for fibroblast adhesion. And this is one experiment that we've done. This is just checking adhesion. Adhesion will tell you nothing about how the cells feel on a surface. If you want to know something about how the cells feel on a surface, you actually check proliferation, meaning doubling of the cells if they divide and so on. And this is an experiment here. Of course, such an experiment takes several days. And as you can see, that the proliferation is accompanied, of course, with adhesion. If you don't have adhesion, you cannot have proliferation. So meaning the nonwovens with the low diameters, with low adhesion, they also have here the low proliferation rate. And the higher the adhesion, also the higher the proliferation. This is not necessary for all the materials. You can have materials that do have very good adhesion, but no proliferation, because the cells, although they bind well, they don't like it. But here, so that's the positive control again. Here we see that in case of the 700 nanometer nonwovens, we do have also a very good proliferation, which is almost identical to the proliferation on this control plate. And here you see an image of a nonwoven made of 500 nanometer fibers, and you see that the cells nicely spread on the surface, and they really have a very nice grip onto that surface. So, just one thing. It's the same protein as in the film. On the film, the fibroblasts don't like it very much. They don't adhere, and they do not proliferate at all. And now just changing the topography from a film to a nonwoven actually increases adhesion and also makes now a surface where the cells can proliferate. Yeah. No, it's diameter. I haven't the picture here. So we just published that 2 weeks ago. Well, we clearly can show what the cells do. I unfortunately don't have the image, but I draw it on the board. So we imaged the cells by electron microscopy. So these are the fibers, and then there are the cells sitting on top. So this is the cell. What the cells do, actually, they try to grip around the fibers. And this is what we see. So if this is the fiber, actually a cell grows literally around such a fiber and gets then in contact with the next cell. If the fiber diameter is too small, actually they don't feel it very well. It's like if you try to hang on a rope which is very thin. Although you can take it, catch it, I mean, you're a climber. So if you have a very thin rope, although you can hold it, you can't grip on very, very strongly, and you slide through. And the same as if it's too thick, right? You can also grip on a thick rope, but again, the grip is not good. So a perfect rope has the size that you can put in one hand, and then you can pull. And it's exactly the same here. Because, as I told you, there's always an interaction between the extracellular space and the cytoskeleton. So if they have a grip outside, they also can direct the cytoskeleton inside. And they pull, literally they pull on the surfaces and on the materials. And therefore also elasticity plays quite a dramatic role. And therefore there is really a very distinct influence of fiber diameter, because pore sizes here are in the micrometer range. They're in all cases quite high. This has nothing to do with the pore sizes, because you can even make as a non-woven with thin fibers that has almost zero pore sizes if you just put a lot of them on top. That of course will also then influence the cell adhesion. But in this case we worked with very large pores, and therefore we were very much convinced that what we see here is strictly dependent on the diameter of the fiber because of this gripping thing. And the same is actually true if then the fibers get bigger, then the cells don't like it very much anymore. So we have here between, let's say, 600 and 1,000 nanometers, like the perfect diameters. Because here they can grip around. I mean, it's like if you want to be Spider-Man and run up a flat wall, you won't be able to do that. But if you have a rope, you can go up, right? And this is why people use silk fibers, as guidance for nerve cells and so on. I mean, they need something to hold on, and then actually they get also an axial growth, and they can get a direction and so on. So let's get back to the film. So now I've shown you something what we try to do to improve cell binding, which is important for tissue regeneration. But now I want to go back to the films. And now I want to show you something that you could do with silk films in terms of tissue engineering or biomedical approaches, although the cells do not bind well. And that is sort of the problem that we got confronted to. So some clinical doctors came to me and said, well, we do have a problem in the clinics. And the problem is silicone breast implants. And it's not the problem that you probably heard in the news in the last year from this France company that they just put industry silicone in the implants, which is highly toxic. No, it's a problem that comes with cancer patients. So if female patients have mammocarcinoma and they have to remove the entire breast, actually there's no tissue left. So in order to remodel a breast, they very often take silicone implants, but because there is nothing surrounding it, there is a huge complication because typically the new growing tissue will recognize the silicone as a foreign material. And what a body does if it recognizes a foreign material, well, A, it tries to remove it. We saw that in the wound recovery. Of course, the silicone cannot be uptaken by phagocytosis and cannot be degraded with proteases. So if nothing happens in the first 2 weeks and this thing still stays there, then actually the body has another trick. It just encapsulates it. So it sort of shields it so that the body has no longer contact to such an implant. And actually that takes place in, I think, like 12% of the patients with such implants that these implants are encapsulated. So we call that fibrosis. Actually what happens is cartilage is forming surrounding it, very tight cartilage, and you even can see that then from outside because this implant is no longer movable. So it's getting stiff, like a rock. It hurts, and it often inflames. And once it's inflaming, then it has to be removed. And this is like 3 months after the first surgery that they have to remove the whole thing again. So you get something remodeled and then they have to remove it 3 months later. So this is a big psychological problem for the patients, and there's so far no cure. And again, we're talking about cancer patients. It's not about beauty surgeries, because there you typically don't have the problem, because you do not regrow the entire tissue. So what you do there is you just take the implant and typically you put it under the muscle. Not much happens there. I mean, sometimes you also have fibrosis, but this is not that big of an issue. But for cancer patients, it's a big issue. So the idea was, can we actually create a surface that is tolerated by the body without having, an ingrow, because you do not want to have tissue growing on the silicon and, even go in. And because if you destroy then the silicon membrane, probably all the silicon comes out, and this is also no good. So you need a sort of a surface that's biotolerable, so cells are sort of okay. They don't see it as a foreign material, but they should also not, make tissue out of it. So that was sort of the idea. So we could show that we can nicely coat these silicon implants. So we do that by dip coating. That's another technique to make films. We did lots of in vitro tests. And then, of course, the next step was to go into animals. And here you see the animal model. It's Sprague Dawley rats. And they get the implant here in the back. So they get a lesion, and then the implant is implanted here. Of course, the implant is not a human implant, but it's the same material, made the same way, just smaller. It's about 2 by 1 centimeters. So the silk film that we generate surrounding these implants is roughly 5 microns. And what we did, we implanted it into rats, and then of course we checked all the parameters of the rats over time, take their blood and measure if there's an immune response, and we saw no immune response, no toxicity, no any kind of other irritation. And then of course we started to see what happens after 3 months, after 6 months, after 9 months, after 12 months, and we did quite a lot of animals, and there was a very intriguing finding. Well, one finding was that we saw a dramatic reduction of the fibrosis, meaning no encapsulation. Here you see just 2 out of 45 whatever animals. This is how typically such an implant looks like after 3 months. And 3 months is sort of like the critical time point. I mean, typically fibrosis occurs in the first 3 months. And what we can see here is that it's really hard for the surgeon to even find the implant. So this is really hard like a rock. I mean, if you take the implant, or if you just push on the rat, you can't move the implant anymore. It's really very tightly packed in that cartilage surrounding. And he really has to cut deep to either see the implant, which would be down there. So it's really coated with a thick collagen cartilage layer. And this is the difference. These are the silk-coated implants. As you can see, he just had to make a lesion in the skin, and immediately he saw the implants. And even from outside, even without hurting the rats, you could easily move them around. So they were not coated by a capsule. There was no fibrosis going on. Even there was a new tissue surrounding it, which was vascularized, which means, yes, it's a very well-tolerated surface, and you see there's not much of an interaction. You still can remove it, so this is exactly what we want to have. It was counterproductive for tissue engineering, where we want to grow tissue, right? Because the cells adhere and then they detach again. But here it's good. I mean, the cells like it. They don't do anything against it, but they really don't want to grow into it. So this actually is one indication that coating the silicon implant makes the implant much more tolerable. Of course, rats are very specific model, so therefore we have now to go to the next level. Next level would be go into pigs, because pigs have an immune system that is more closely related to humans. And then you get much more insights. And then, of course, in pigs, you can use real size implants. There are, of course, some differences then in processing and so on. And this is something that we're preparing currently. But this was only the one interesting thing. There was another interesting thing, and this was really intriguing, and we didn't think about that to begin with. Has any one of you ever seen a silicon implant or hold it in the hand? Probably not. Well, I did neither before we started that. But we got the implants, and we just unpacked them and just put it on the shelf. And like after a few days, they're getting slippery, slimy. Why? They're not tight. So there's always unbound silicone oil that actually removes through that silicone membrane all the time, also in the body. So the surgeon said, well, we can see, if we make histological cuts, we can see the silicone oil, surrounding the implant. And even like if you have human patients and they die, and you do the pathology, they can even find then the silicone like in the knee joints, in the hip joints. This is where the silicone accumulates. Because the silicon cannot be removed from the body, so the silicone oil. So you find this even after like 30, 40 years, even if you had an implant like in your younger years and then you remove it, when you die, the silicon still will be there, will be not degraded. So what we found with having these silk coatings, and again, remember, silk is amphiphilic, the silicon surface is quite hydrophobic. So what happens here is that the silk, again, like with the capsules, brings all the hydrophobic surfaces towards the silicon. And this somewhat interacts also with the unbound silicone oil. And actually, you make kind of an oil layer underneath the silk film, but the oil cannot pass now the silk membrane, because the silk membrane on the other side is hydrophilic, right? So you have sort of like a repulsion. mean, the silicon membrane is hydrophobic inside and outside, so therefore you have the diffusion, the free diffusion over the membrane. The silk membrane is hydrophobic inside. You have the accumulation of the silicon oil, but it's hydrophilic outside, so you don't have a lot of transmission or even no. And the surgeons were really surprised because they could not detect or just detect very little of silicon oil surrounding the implant. And this is another nice feature, and this is something we try and, to push now to the next level to see what we could do there. So, of course, we did histological measurements and so on. And again, this is now just some data to show you that indeed the coating of the silicon implants have some good effects on the cells. Let's start with a graph here. So that is an implant without the silk coating. And here we just counted the inflammatory cells surrounding the implant. And you see here a drop by, more than 1/3 once we have the implant coated with the silk protein. Of course, we never get no inflammatory cells, but it's a severe drop. And the same is true for fibroblasts, which are indicative of making collagen, right? And collagen is making this fibrosis. So if you have less cells making collagen, of course, the chances that you get fibrosis is much lower. And there's also less, not that much, but less collagenous connective tissue surrounding it. So these are clear facts that our, not only like the visual stuff, but facts that our coating really improves the biocompatibility. And here is some other showing. So these are stained tissues, and the staining was against CD68 macrophages. So typically, if a foreign material comes into the body, macrophages, as I told you, are induced to make phagocytosis, to remove the foreign stuff. And this is the case here. So all the dark spots, these are all macrophages. You see quite a lot of them. And they even actually, so this is like the interface here would have been the implant. So this is the interface between the normal tissue and the implant. And as you can see, there's a huge accumulation of the macrophages at that interface, and it's even that severe that we have a lift off of the tissue. So the tissue is no longer intact, so you have sort of like a failure in the tissue. And that's the tissue surrounding the silk-coated implant. Of course, you do have some macrophages here, but this is just a normal sort of distribution. The tissue is fine. There's not much of a lift-off going on. The lift-off here is actually coming from this collagenous connective tissue. So this is the beginning of fibrosis, right? So this is the capsule that will then surround the implant, and you see it's lifting off the rest of the tissue. And this is not the case here with the with the silk coated implants. So this is after 3 months. But we also did it after 6 months, 9 months, 12 months. There's not much of change because typically fibrosis takes place within the first 3 months. Of course, what is then different and what we now, we haven't analyzed all the data yet. Of course, there will be a difference in removal of the silk coatings. We have seen that the coating is intact after 3 months, after 6 months. So the silk proteins are stable. We don't know about the 9-month and 12-month time points so far. But we guess that the silk film is still there. So it's quite stable because there's not much of a wound recovery response and remodeling going on at that interface. Okay, so this is pretty much what I wanted to tell you. And of course, I would like to use the final lecture also to thank the people. And this is the flock. People working in my group. That's my secretary right here. And so I'm back here. And here are some technicians and diploma students, but most of them are PhD students. Most of them, let's say 70% work on silk and silk processing and materials, but the other 30% work on other molecules I haven't talked to about, mussels, for instance, and they also have intriguing fibers. So many thanks to them, because without them, of course, we couldn't do that kind of research. There's quite a list of alumni from the lab, and of course, we do have quite some collaborators, polymer chemists. This is the NMR specialist, Horst Kessler. We made the structures of the termini with Andreas Bausch. He's a physicist also in Munich, with whom we started microfluidic stuff. Gerhard Winter, he's a pharmacologist, because of course we have no idea about drugs, so we need an expert on that, so that's him. And we have a very nice collaboration with Dave Kaplan, with whom I've been in contact since 10 years, and who was, well, I think the only one who encouraged me 10 years ago to get the whole thing started. All the others, even the silk people, said, we know everything. So far about the collaboration partners. And then, of course, we do have quite some funding, Bavarian funding, German funding, industrial funding, funding from the US. Very grateful for that. This is our company, so if you want to know more about the company, please check www.msilk.com. So they have a nice homepage telling you about their activities. And of course, there's furthermore, much more supporting companies. And please feel free to also check our homepage, www.fiberlab.de, where you see much more about the activities, about the people, about the projects that we do, about the press, movies, and so on. Most of the movies are in German, but there's some nice stuff from the BBC where you also can see it in English. And finally, of course, that's sort of my final take-home message. There are some really fascinating materials out there, and this is sort of like our main theme. We try to, well, study these, and of course we relate on also studies from groups like yours, because we will not have time and capacities and knowledge to really understand everything that's going on there. We are more at the transition zone, you know, to get or to take that knowledge, to make now new materials based on what we know from nature. So this is sort of bio-inspired materials research. And then we also try to understand how to process the materials, well, natural processing in forms of fibers here, but also, of course, other processing forms. And then, hopefully, we will get some applications soon, and then I think I talked a little bit about that, what we intend to do technically, but also in terms of biomedical research. With this, I would like to end, and of course, I'm still open for questions. Thank you. It's a pleasure. That's good. I mean, that was the intention. You're very welcome.